Good afternoon. Okay, so we are going to get started with the dissertation defense of Bradley Lusk. So I'll say a few things about Brad that you might or might not know. He is actually from here, from Arizona. He did his BS in biology in 2009, and then within it did a certificate in philosophy of science, which I thought was kind of interesting. In 2009, he started the biological design program. And that was second year of the biological design program, or
second or third, yeah.
Depends on how you count. And he started with the Science Foundation Arizona Fellowship back then. The biological design program is quite interesting, and it's gone through a few evolutions, as you know, if you work around the students. And Brad is an interesting part of this, program. He has right now 2 publications and a third one almost accepted, hopefully, and a few others that we're working on. He's presented. I listed here places where he's presented, so you get an idea of the interdisciplinary part of Brad's work at the Association of Environmental Engineering and Science Professors Conference, at the International Society for Microbial Electrochemistry and Technology Conference, and the Association for American Society for Microbiology Conference. So kind of a mixture of microbiology, engineering, and somewhere in everything kind of in between. He also went to Kuwait ESM, a university in Pakistan, but that's kind of part of one of those names at the bottom of Brad's name there. So that's kind of the science background related to Brad. He has a second phase of his life, which is related to education. Since 2005, he's been part of the board of Camp Sparky, which is a elementary outreach program. And he was the president. You're still the president, or no anymore? Last year, he was the president of Camp Sparky. He's had a few talks, and I think this is also related to the biological design program to push you to do some outreach. He's had a few talks at Comic-Con Science Cafe, that's part of the Arizona Science Center. And he's been mentor of quite a few people. If you look down there, there are some high school students there. There are some a few visiting scholars, which are co-authors of some of the papers that we're writing right now. So that basically adds up Brad in general. So what is it that Brad is going to talk to us about? So being from Arizona, Brad likes the heat. And so he worked with a thermophilic bacteria during his time here. For those of you that come from outside our team, we work with bacteria that produce electricity. So that means that Brad needs to make these interesting combinations of electrochemistry and microbiology, which are kind of difficult to put all inside an incubator at 60 degrees that you cannot touch because you burn your hands. So having said that, I'll leave it to Brad to tell us his story of his PhD.
Okay. Thank you, Cesar, for that introduction. Okay, so my name is Brad, and today I'm going to talk to you about thermophilic microbial electrochemical cells. I just want to let everyone know that this research has taken place at ASU over the last 6.5 years, and it is much a feat of interdisciplinary science as it is a feat of kind of interdisciplinary just work in general, or I should say multicultural, international, and also multi-educational. So as Cesar mentioned, I have worked with high school students, master's students, undergrads, PhD students, and doctors over the course of this research. And I wanted to acknowledge some of the people that have directly impacted my research before I continue. And so we have Prafet Parameswaran, Sudeep Popat, Bruce Rittman, Cesar Torres, Rosa Krochmalnik-Brown, Kaiser Farid, who I worked with in the lab. He's the individual I visited in Pakistan. Gaurav Albal was the first high school student I interned. Alexandra Collin is a master's student from France that was with us for about a year. Carlos Alvarado was another high school intern that I had. Alexander Zevin is someone who was previously in the lab. John Badalamenti, Daniel Bond, Tyson Bree, Diane Hagner, Young So Lee, and Daewoo King, who were actually responsible for doing 2 of my rotations the first year that I was here. All of these people have played a very, very large role in getting this research kind of off the ground and moving. I also want to let everyone know that this is being broadcast live online right now on YouTube. And I actually just found out that someone that I know, her elementary school, 5th grade elementary school class is actually watching this right now. So keep that in mind when you're asking questions as well. And I also have a joke in here. Okay. Okay. So today we're going to be learning about single-celled organisms. Now, all of us should care about single-celled organisms because all of us spent 30 minutes as a single cell. The microorganisms we're going to talk about are called bacteria. Now, bacteria are important because bacteria are ubiquitous. The bacteria associated with our bodies, for example, the cells, outnumber our cells 10 to 1. The genes associated with those bacteria outnumber our genes by 100 to 1. And bacteria are the catalysts of the future. The bacteria that I research are capable of catalyzing the transference of energy in organic waste to electrical energy or hydrogen that we can then harness and use as fuels. And they can do this because they're what we call exoelectrogens or anode respiring bacteria. That is, these bacteria are capable of breathing metal. So what does that mean? So I'll give you all an example that you might be more familiar with. And that is, I assume that all of us have enjoyed an apple at least one point in our lives, or know of people who do. And why do we eat those apples? Well, those apples are nutritious, delicious, but I want to focus on the energy that those apples provide for us. Those apples provide us energy in the form of electrons. Now, I know it might seem odd to think about food as having electrons or electricity in it, but that's actually kind of how it works. So, as we digest the apple, our body, we break it down into small apple pieces, and our body ingests those pieces, and from those pieces, we extract energy, right? This energy eventually gets to what we call the electron transport chain in our cells. And here it is, the electron shows up in our cells. What this electron does is it travels through a series of proteins in order to pump protons across a membrane to make a proton gradient. Now everyone take a deep breath. There's oxygen in the air we breathe. Why do we breathe that oxygen? Well, I'm about to show you. So we use this proton gradient and put it through a pump called ATP synthase, and we generate ATP from something called ADP and phosphorus. And ATP, or adenosine triphosphate, is the energy that our cells use so that we can do work and survive. ATP is the fuel of the cell. After we generate ATP, we now have this electron sitting around, and we need to get that electron outside of our cell. We don't want a bunch of negative charge hanging around. So how do we do that? Well, we can take those protons and that electron, combine it with that oxygen, and we can reduce that oxygen to generate water. Now, how many of you have seen your breath on a cold day? We have a lot of water vapor in our breath. We also perspire. This is where some of that water comes from. This is why we breathe oxygen. We need a terminal electron acceptor. So now imagine you're a microorganism and you're poisoned by oxygen. That is to say, you're what's called an obligate anaerobe. That means you can't survive in the presence of oxygen. So you can't perform respiration using oxygen. So what some microorganisms do is they use their electron donor as an electron acceptor. This process is called fermentation. Yeast are very good at fermentation. They take the sugar that we give them, and they produce ethanol from that sugar, in order to generate energy for their cells. Anyone who's enjoyed a good drink knows that this is true. And anyone who's interested can join me at Four Peaks after this. I'll be there around 4:30 or 5 in happy hours until 6. But let's imagine that we don't want to do fermentation, we want to do respiration, because in general, when cells perform respiration, they can generate more energy per molecule that's coming in. Now, we still have this electron, and for simplified reasons, I kind of have this drawn as the same. The exact process in bacteria varies by bacterium and is actually different from what's happening in our cells. However, we still have a series of either quinones or cytochromes that are moving these electrons moving the electrons in order to generate a proton gradient. So now we have an electron, we're moving it, we're generating a proton gradient, we're taking those protons, we're passing them through a pump, we're making ATP for our cells. So far everything looks fairly similar. But now we have this electron laying around and we have no oxygen to take this electron. So what these cells have adapted to do, or what these cells do, is they have membranes, they have proteins within their membranes that are called cytochromes. And the electrons can actually be transported outside of the cell using these. And what we have here, instead of oxygen, is we have an external electron acceptor, and this is an insoluble metal. In this case, we can just say this is an iron oxide. So now we're putting electrons onto our electron acceptor, and we're also moving protons outside of our cell. Every time we move out a negative charge, we have to move out a positive charge with it. This is important to keep in mind because now our cells can keep doing respiration, they can keep generating ATP. However, if they do it a lot, they're going to generate a lot of protons outside of their cell. So when we put a lot of protons outside of our cell, what happens is the pH goes down. So what that means is our cell is now in acidic conditions. pH, after all, is a measure, in this case, is a measure of the protons that are in the media. More protons, lower pH, more acidic. Less protons, higher pH, more basic. If we have too many protons outside of our cell, we're going to start shutting off this pathway. We're going to start inhibiting this pathway. So now our cells can't grow anymore. They can't respire. This is an example here, taken from a paper by an individual who's in our lab, Andrew Marcus, and I think Cesar is also on this paper, where we're showing, here we have an insoluble metal, in this case it's an anode, and I'll get into how this works later. And in this gray area here and over here, we have bacteria that are growing off of that metal. And what this graph is showing us is that at high pH, throughout the biofilm, or throughout where these bacteria are growing, all the bacteria are happy. They can all perform the respiration that they need to survive. If we look over on the left here, we can see the pH is really, really low, and we have this hashed gray area. This hashed gray area is where these bacteria are inhibited, or they can't grow, they can't survive. Hence, frowny face over here. So what can bacteria do? What can we do as obligate anaerobes now? And by the way, I should mention that not all exoelectrogens or anode respiring bacteria are obligate anaerobes, but the ones I'm researching are. So what can these bacteria do to adapt? Well, we know this pathway is off. Well, we also know that bacteria can use several electron transport and electron transport chains simultaneously, and also can send electrons out of their cell using different electron transfer mechanisms. So what we can do is have bacteria that have several of these different processes going on. That way they can kind of adapt as pH is changing in the natural world. However, if we keep lowering the pH, we're eventually going to inhibit all of these pathways. So how can we fix this? Well, we can add something called bicarbonate. Now, all of you are probably familiar with bicarbonate and you don't even know it, because bicarbonate is essentially just baking soda. So we can add in bicarbonate, and bicarbonate acts as a taxi that can swoop in. It can pick up those protons. It can generate H2CO3. And then it can move those protons outside of the biofilm. And if we add enough of that bicarbonate, we can start to get rid of those protons. We can start to neutralize the pH. When we do this, these pathways can start turning back on, and we can start, once again, sending electrons out. But how do those electrons get out of the cell in the first place? What do these pathways actually look like? So now I want to focus on kind of what the pathways look like physiologically within bacteria. And so this graph, or this picture, looks very similar to the one I just showed you. This is what it looks like in what's called a gram-negative bacterium. You have two membranes, an inner membrane and outer membrane, with a periplasm in between. And you have a peptidoglycan layer that is so small in gram-negatives that it's often not even considered when people draw pictures. Hence, it's not even in this picture. And what you can see is there's a series of proteins and cytochromes that are associated with getting that electron outside of the cell. The bacteria that I'm using are what's called gram-positive bacteria. Now, this is significant because not a lot of research has been done in this field using gram-positive bacteria. And gram-positive bacteria are a lot different than gram-negative bacteria. Gram-positive bacteria have a very, very thick, what's called a peptidoglycan layer. Now, this peptidoglycan layer can be 10 to 40 times thicker than what's present in a gram-negative. And why do we care? Well, we care because this mechanism for getting electrons out of this could be fundamentally different than the mechanism over here because we have a much different cellular structure and framework. And for those of you who are in the know, this slide is still MAC A pledge approved. You're welcome, John. Okay, so now we'll go back to this picture. And once again, this is what a gram-negative looks like. This is what we went over. But what I'm looking at is a gram-positive. So it looks more like this. So we can see now how this might work very different. We might either have electrons that are going across cytochromes as we did before, or proteins that are embedded within the peptidoglycan layer. Or some people have predicted that we might have the protons going out through what's called teichoic acids, which have been linked to metal binding in gram-positive bacteria. So we're interested in figuring out how exactly our bacteria, our gram-positives, are doing this. And so let's look at how, once the electrons actually get outside of the bacteria, how are they actually going to the metal? And so we know of 3 different ways that the electrons can get to the metal or the anode that's outside of the cell. One of those ways is called direct electron transport and transfer, and that is where a bacterium is literally attached directly to the metal or the anode. Another one is called long-range direct electron transfer and transport, and that is where there is a network of extracellular appendages that are coming out of the bacterium that are sending the electrons to the metal or anode. And we also know of bacteria that have redox shuttles, so shuttles that are reduced at the bacterium, go to the metal or the anode, become oxidized, travel back to the cell and get reduced again, and so forth. The bacteria that we're looking at today, at least the ones called Thermincola ferriacetica, which I'm going to tell you more about a little bit later, they're using what we believe to be this pathway. However, it's important to consider the genetic lineage of these bacteria when we're considering the pathways that are available. And so what I'm showing you here is that we have what's called our last universal common ancestor, which is Luca here. I have no idea what this looked like. Some goo, I don't know. And up here we have eukaryotes, that's us, and archaea, which are other single-celled microorganisms that I'm not going to talk about today. What we're concerned about is this lineage down here, the bacterial lineage, because we're talking about bacteria. And this big black dot right here represents the last universal bacterial common ancestor. So this is the last ancestor that was common to all bacteria. And all of the bacteria that have been studied before, or most of them, belong down here in gamma proteobacteria, delta proteobacteria, etc. A lot of them are gram-negatives. The bacteria that I'm looking at are gram-positives that belong to what's called the Firmicutes phylum. And we can see that the Firmicutes phylum, these bacteria did not beget these bacteria. What this means is we might have genetic similarity here in this common ancestor that ended up in both of these, or we could have what's called convergent evolution, and that is we had a group of bacteria that evolved a mechanism for doing this here, and a group of bacteria that evolved a mechanism for doing dissimilatory metal reduction here. And those two processes perform the same function, but they aren't from the same genetic lineage. To give you an excellent example of that, you can think about the wings of a bird versus the wings of a bat versus the wings of a fly. They all do the same thing, but they don't have the same genetic lineage. So now let's also consider this, because some of you might be thinking, oh, respiring metals, that's weird. Why would you do that? But let's put it into perspective. So here we have the Earth is formed 4.5 billion years ago. And out of the formation of the Earth, we have the first prokaryotes, the first single-celled the Earth was formed about 5 billion years ago. The first prokaryotes, single-celled microorganisms, about 3.5 billion years ago. And what we call the, the oxygen, formation of oxygen is about 2.5 billion years ago. So we have single-cell microorganisms from 3.5 billion years to oxygen at 2.5 billion years. And we have 1 billion years in between for microorganisms to evolve without the presence of oxygen. We also know that the origins of life were thermophilic. That is, the origins of life like high temperatures, something over 60 degrees centigrade or over 140 degrees Fahrenheit for all of us in the United States. And why would these microorganisms evolve in high temperature conditions? Well, let's think about the old Earth. Let's think about the Earth when it was first forming. The surface was scorched by UV radiation, and UV radiation is very dangerous to life. We all know if we spend too much time in the sun, we can get cancer or a sunburn. We still use UV radiation to sterilize stuff today. But underneath the water, the UV radiation can't penetrate as far. And underneath the water by geothermal vents, there's a lot of energy that can be used to catalyze reactions, energy in the form of heat. And we also know by these geothermal vents, we have high amounts of metal oxides, iron oxides, and humic substances. So it only seems to make sense that these microorganisms would evolve in thermophilic conditions with a lot of metal oxides around and figure out a way to perform respiration using what was available to them. So how do we study these microorganisms? Well, we study these microorganisms using something called a microbial electrochemical cell. So what's a microbial electrochemical cell? Microbial electrochemical cell is essentially a bacterial battery. We have an anode, and we have a cathode, and we have a membrane that kind of acts as a salt bridge. In the anode, the bacteria are capable of oxidizing reduced organic contaminants, taking those electrons and transferring them to an anode. So let me show you kind of how this is working with gram-positives. So we have gram-positive microorganism. We have an external electron scepter, which in this case is an iron oxide. We remove that iron oxide and we put it in an anode instead and connect it to a wire. The bacteria then perform respiration and they pass electrons through this anode and then out through this wire. Also, remember, we're always pumping protons every time we move an electron. These electrons then go to what we use in the lab. It's called the potentiostat. And what this potentiostat does is it takes that electron, it adds a small voltage, and it sends it back to the microbial electrochemical cell, because we're using a specific kind of microbial electrochemical cell, which I'll tell you about in the next slide. And it also generates, a signal to send to the computer so that we can look at all this using our GUI, or our graphic user interface. Now, the kind of electrochemical cell we're using is called a microbial electrolysis cell. So we take that electron, we put it in the cathode, and we use it to split water and generate hydrogen, hence electrolysis. We're using electrons to lyse water and generate hydrogen. We can then take that message from the potentiostat and we can display it on a computer. So what are we doing here? Well, we have bacteria. They're growing on an anode in thick biofilms. This red that you see here is actually a bacterial biofilm that's grown on the anode. And those bacteria are passing electrons onto the anode, which is going to the potentiostat, which is going to the computer, which is in broadcast online, and I can communicate with my bacteria using my cell phone. That's what we're doing. How do these bacteria get on this anode? Let me give you an analogy. If we were going to be jerks, we could go to a pool that had a bunch of people training to be scuba divers in it, and we could rip off all their scuba gear and just troll them. Where do you think all those scuba divers would go? To the surface of the water. And all those scuba divers would go to the surface of the water because that's where their oxygen is. That's where their terminal electron acceptor is. So what happens when I put these bacteria, when I submerge them into a bunch of water and I don't give them any scuba gear? They go to where their electron acceptor is. They swim to the anode. And they're quite happy living there. As a matter of fact, as they're living on the anode and growing, they're sending electrons to that anode and generating a current. And as they generate that current, the potentiostat is monitoring that current. And by monitoring that current, we can actually track the bacterial growth over time. So this is what some of you might recognize as the lag phase, or the phase that it takes the bacteria to start growing. This is what's called the log or growth phase. This is where the bacteria are starting to grow exponentially. And this is what's called steady state. This is when the bacteria are done growing. If we were to carry this out far enough, we would actually start to see what's called the decay phase, or where the bacteria begin to decay. And we can see this in real time. This is monitoring in 2-minute intervals over 8 days. We could monitor in 1-second intervals if we wanted to, but we get a data file so big we wouldn't be able to process it. But this is essentially how the bacteria are now communicating with me. They're letting me know if they're happy. These ones look pretty happy. We can also do something that's called cyclic voltammetry. And what we can do when we do this is we can actually sweep the potentials of the anode. As we change the potential of the anode, we're changing the energetics of the reaction that's happening within the biofilm. And as we make the potential more positive, the biofilm is able to generate more current. And as a response, you can see the current goes up as the potential goes positive, and the current goes down as the potential goes negative. So what do we call it where the current is the highest or the maximum it can be? Well, for shorthand, we say current is J, and where the J is maximized, we just say that's J max. And then if we want to look for the midpoint potential of this reaction, which is about half of what J max is, we just call that 1/2 J max. Or we can call it the EKA. And what this midpoint potential is telling us, it's telling us the potential at which the process that's responsible for doing the electron transfer outside of the cell is half oxidized and half reduced. We also have something called the Nernst-Minot equation, and you can kind of see a fit here. It kind of fits. And what the Nernst-Minot equation is telling us essentially is whether or not this is one process that passes one electron happening, or if maybe there's more than one process going on. So if it fits Nernst-Minot very well, we can say, okay, this is one process. And if it doesn't, we can say, okay, maybe there's something more to it. Maybe we should research this further. There's another way that we can probe or look for different redox active proteins, and that is we could just look at the genetic information. We could look at the DNA and we could say, hey, if there's something there that codes for a cytochrome, then that cytochrome could be expressed. It's likely that it's expressed. I mean, it's possible that it isn't, but it's there. It's probably there for a reason. It's probably there to get expressed. So there wasn't a lot of genetic information available for Thermincola ferriacetica, so I decided we should grow this and extract the DNA, and I sent it off to John and Daniel Bond, and they took that DNA and they made, they multiplied it randomly. They took those random sets of DNA and put them into contigs and then assembled that into a whole circular chromosome. So we have mapped the entire genome. We've made a draft genome of this entire bacterium. We know it's about 3.19 million base pairs, and it has about 35 of these C-type cytochromes. So now we have Thermincola ferriacetica, finally, the star. Here he is, or here she is. Here it is. We know Thermincola ferriacetica respires metal. We know it's a gram-positive bacterium at part of the Firmicutes phylum. We know that it's thermophilic. It operates at 60 degrees centigrade. We know that it has a pH range of 5.2 to 8.3, and we know that because of the data I'm about to show you. We know that it has 35 multiheme C-type cytochromes, and we also know that it consumes acetate. So acetate is something that we're also very familiar with, or a close form of it. This is just vinegar. So we think about the apple earlier. Let's just say this is apple vinegar. So we're feeding it apple vinegar. And down here I have some pictures of it. So this is kind of what it looks like as a cross-section growing on an anode. This is what an individual cell of Thermincola ferriacetica looks like. This is it growing again in just kind of what we call EPS, or extracellular polymeric substances. It's just kind of all bound together by this glue. And this is a very, very far away view of the biofilm. It just kind of looks like maybe like mold growing on an anode, because we're so far out, we can't see the individual cells. So the first thing I wanted to do is we wanted to probe whether or not this bacterium was inhibited by pH. According to the introduction, the bacterium should eventually be inhibited by pH, and we just want to know what is that pH range for this microorganism. And what we can see here is at a neutral pH, we're at about 80% of the current density that we can get at a pH around 8.3. 8.3, which is represented by purple here, is where we saw the highest current density. And what this graph is showing you, pH 8, what this graph is showing you is current density versus the current density at pH 8. So we can see at pH 7, we have about 80% of the current density is at pH 8, and around pH 5 or 5.2, we have about 20% of the current density versus pH 8. So what we can tell is that this bacterium is limited by pH at low pH, and after pH 7, after neutral pH, that limitation becomes a little less significant. And we want to figure out, or I want to figure out, why. What's going on? So the first thing I do is I want to probe to see what pathways are there. What are the pathways doing that are getting the electrons outside of this cell, and what ones could possibly be inhibited by a low pH? So the first thing we do is I scan at a neutral pH, and we can see what appears to be one pathway. It has an okay fit to Nernst-Minot, but not quite right. And so we think there might be something up. We look at the apparent midpoint potential. This is about minus 250 millivolts. I then lower the pH to about 5 and do another CV scan. And interestingly, I see something that looks much different. So what's happening when I'm doing a CV scan? Well, if we remember earlier, as I'm scanning forward, as I'm increasing the voltage, the bacteria are producing more and more current. And we know that when we produce more current, we produce more protons. And we know if we produce more protons, we lower the pH. So we can see in a 6.9 setting with 50 millimolar bicarbonate, remember the taxis, we have enough bicarbonate in a high enough pH so that we don't see any inhibition. The forward looks the same as the back. When we go to the pH 5.2 condition, as we're moving forward and generating protons, we're actually lowering the pH enough to where when we scan backwards, we see that we have a lower current than we did on the way forward. So what's going on here? We're going to get into that soon. We know this EKA is about minus 190 millivolts. And we can see that there's a potential shift of the apparent EKA about 60 millivolts, which is actually what we would expect to happen as we lower pH, because we're also changing the thermodynamics of the acetate that we're feeding this reactor. The next thing we want to compare is the high pH condition, and we do that, and it looks, again, like there's one or maybe a few more processes going on. We can't really tell. The forward and reverse look the same, but we can also see that there's a 20 millivolt negative shift from pH 7 to pH 8. And this is also associated with the change in pH, changing the thermodynamics of what we're feeding it. And as you can see here, what we're feeding it, acetate, has a set potential that is changing with the pH. And as we're changing that set potential, the microorganisms are able to shift their potential or adapting so that they can still generate ATP from this process. So what do I think is happening up at this really, really low pH condition? Well, let's look at the derivative curves of these experiments. And basically what we're doing with the derivative curve is we're just kind of looking at the rate of change over time. And we expect to see the greatest rate right around the midpoint potential. And that's what we see on the forward, and then underneath the hash line is the reverse. And remember, this is the pH 7 condition. Everything looks the same. What appears to be one midpoint potential. And there appears to be a very, very wide range that this midpoint potential covers, about 250 millivolts. Now, when we go to the pH 5 condition, we can see a much different story. There appears to be two midpoint potentials on the way forward, but only one on the way back. And we also notice on the way back, it's only about 200 millivolts. So what could be happening here? Well, what could be happening here is as we're shifting forward, we have a pH around 5.2. We're lowering that pH, and as we come back, we're actually inhibiting one of those pathways. And as a result, we only see one We only see one peak on the way back instead of two. And also, we see a smaller range in the midpoint value down here because those two aren't overlapping anymore. So if we look at the high pH condition again, it looks the same as the neutral pH condition. So essentially what we're seeing here is it looks like at very, very low pH, our biofilm is experiencing some inhibition, potentially by shutting off a pathway that it's using to take the electrons outside of its cell. So now let's go back to the cartoon and think about what's happening. So at pH 6.9, it could be that we have two different pathways and the bacteria is happy, it's chugging along, it's producing current, it's making ATP. At the pH 5.2 condition, once again, we're using two pathways. This is the way forward. Maybe one of these pathways is starting to get a little bit inhibited. We're driving that pH lower and lower and lower as we're pumping more protons across. And what's happening is on the way back, we're actually looking at maybe a pH that's a little lower than 5.2. And as a consequence, bam, we shut off that pathway. And we're not seeing as much current production on the way back. We're also seeing a smaller midpoint potential range because now these two ranges aren't overlapping anymore. And we know at 8.3, it should look about the same as at pH 7. So what can we do with this information? Well, now we have the electrochemical information and we have the biological information. So we know that it looks like there's two peaks and we know that there's at least 35 C-type cytochromes. So there could be multiple pathways. And now what we can do is we can take mathematical modeling and we can say, okay, if those two pathways are on and they are working together at the pH 7 or pH 8 condition, we should be able to model that and see. And that's what we've done. So here I have two pH 7 reactors. One was producing 11 amps per meter squared, the other 12, which is represented by the purple and the blue here. I've taken the pH 5 midpoint potentials and I've modeled them to fit these. In other words, this is what one of those pathways would look like, and this is what the other pathway would look like. And I say, okay, if at this pH 7 condition I'm seeing the combination of those two pathways, then I would expect to see this black hash line right here. Oops. And you can see that this black hash line fits the data almost perfectly. So that's telling us that it's very, very likely that there are at least two processes that are happening to get the electrons outside of these cells in these gram-positive bacteria that we know very little about until now. The next thing I wanted to do to kind of confirm this hypothesis is look at how current density changes as I change the bicarbonate buffer concentration. That is to say, when I add more taxis to take the protons out, do I generate more current? And when I take those taxis away, when I take the bicarbonate out, do I generate less current? And that's what we can see here. So at 10 millimolar bicarbonate, we see about 60% of the current that we get at 100 millimolar bicarbonate. And what we can tell from this graph is from 10 to 50 millimolar bicarbonate, there is a statistically significant change in the amount of current that we can produce. After 50 millimolar bicarbonate, statistically, it doesn't matter anymore. So we're reaching another limitation once we get to 50 millimolar bicarbonate. Then instead of going through all those CVs individually, for the sake of time, I'm going to show them all at once. And what we can see now is that 10 is green, 25 is blue, purple is 50, and red is 100 millimolar bicarbonate. And what we can see is at lower bicarbonate buffer concentrations, in other words, at concentrations that will not be able to take as many protons outside of the biofilm, thus the biofilm will be at a lower pH, we can see that we have the same thing occurring. We have what appears to be two peaks at lower pH, and at the red at the higher, or at lower buffer, and at the red at the higher buffer, we see only one. So what might be happening here? If you notice here, this pH says 6.9. However, on the last reaction, we had 50 millimolar bicarbonate. We had more taxis. We had more taxis to take the protons outside of the biofilm. On this condition, we only have 10 millimolar bicarbonate, so we have 5 times less. So the result of that might be that even though in the bulk media, we think we have a pH of 6.9, in the biofilm, we might actually have a pH that's closer to 5 or 5.2. We also know that these biofilms are very, very thick, and because they're very, very thick, we might be able to generate a very large proton gradient within these biofilms. So what this is, it's kind of hard to see, but this is called confocal laser scanning microscopy. And in this, what I can do is visualize these biofilms as they're growing on an anode. So you can imagine there's an anode here and bulk media out here. And now I'm taking a microscope and looking at it coming from this way down. And this is a Z cross section of it. So now we're looking at the thickness of this biofilm. And what we can see here is all the green represents alive bacteria and all the red represents dead bacteria. And we can see that this biofilm, the active portion of this biofilm, is about 60 to 80 microns thick. And I did this several times at several different bicarbonate buffer concentrations, and I saw that at lower bicarbonate buffer, 10 and 25 millimolar, we have about 60 to 80 micron thick biofilms on average. And if we give it more bicarbonate, if we allow for there to be more diffusion of protons out of the biofilm and thus more layers of happy bacteria respiring, we can get thicker biofilms. So at the 50 and 100 millimolar, we have around 150 microns thick biofilms. So very, very thick biofilms. The next thing I did is I wanted to look at how these bacteria change, how their midpoint potential, how their midpoints might change as they're growing. And so what I did is I grew these in a microbial electrochemical cell from 0 to 13 amps per meter squared. And I took a CV at almost every single step between 1 and 13 amps per meter squared. And you can see it looks very beautiful. I just want to show this. It looks so beautiful. I love this. When I saw this, I posted it on Facebook, and Jordan, who's also on my program, was like, "That's so beautiful." And I was like, "Yeah, okay." So what we can do is we can then do the same thing we did before. We can look at the derivative curves of those values, and we see at pH 7.4 at 1 amp per meter squared. So now we're imagining it's a biofilm that is not very thick. It's just starting to grow. We see what appears to be one peak. As our biofilm grows, now we're at 7 amps per meter squared. We're somewhere between 7.4 pH, which is where we started, and 7.0 pH, which is where the pH was at the end of this experiment. And we can see that we're starting to see what appears to be multiple peaks here. And if we take it one step further and we go all the way out to the 13 amp per meter square condition, these peaks are starting to get really, really apparent. And what's happening is now our pH is getting lower. And so we're starting to see those two pathways showing up. We're also seeing a 10 millivolt shift to the right as the pH is changing by about half a pH unit. So once again, as the pH changes, the apparent midpoint potential is also changing. It's exactly what we would expect to happen. The last set of experiments I did with Thermincola ferriacetica is I took out all of the acetate, all of the vinegar, all of the food that it had to get electrons from. The biofilm was already grown on the electrode, and I poised the electrode at different potentials. So now we're in a situation where the bacteria can only get electrons from the electrode or have electrons taken away from it, from the electrode. And what I'm looking at doing there is looking at the inherent value of the pathways, the inherent midpoint potentials of the pathways that are associated with that electron transfer. And what I can see is that, once again, we can see 3 different bumps. We can see one combined bump up here, and then 2 that are kind of separated at the bottom. So in the last part of this, I just want to kind of transfer into, okay, now we know about the really, really cool microbiology that's associated with these bacteria, right? With gram-positive thermophilic bacteria. Now let's start thinking about, okay, now we have all this knowledge, how are we going to use it? How are we going to use it? So with Kaiser, who I introduced earlier at the beginning of the slide, the person from Pakistan, I looked at a different anode-respiring gram-positive bacterium called Thermoanaerobacter pseudoethanolicus. And what's cool about this bacterium is not only does it respire metal, but it also can ferment sugars. Sugars including glucose, xylose, cellobiose. And we know that it's thermophilic and that its pH range is between 5.4 and 8.2. Once again, it's from experiments that I did in the lab. I don't have time to get to all that data today. And also, this one has also been isolated, so it's really easy to order. And you can see with the scanning electron micrographs down here that this is what that bacterium looks like when it's growing on an anode. And it has these drumstick-like appendages coming out of it, which are indicative to what this bacteria looks like when it's growing. Now, why this is significant is because now, instead of having only acetate, which in one review paper called La La Land, we're never going to have a stream of 100% acetate to feed, well, I don't want to say never, but it's going to be unlikely that we can find a stream of 100% acetate to feed to a microbial electrochemical cell so that we can generate hydrogen from it or electricity from it. So what we're interested in looking at here is, well, streams are diverse, so let's see if we can convert diverse wastes into hydrogen or electricity in a microbial electrolysis cell. And what we can see, or current, I should say. And what we can see is if we put in xylose here, which is a more complex sugar, this bacterium ferments it almost instantly and produces a lot of acids. So here we have acetate. And then it can consume that acetate and generate current over time. We can see that it can do the same thing with glucose, once again producing acetate and lactate, and then converting that glucose into current in a reactor. And we can see that it does the same thing with another sugar, cellobiose. It's taking cellobiose, which is a significantly more complex molecule than acetate, and it's turning that into current as well. But what's the ultimate challenge? The ultimate challenge in the microbial electrochemical cell field is to turn solid wastes into hydrogen or electricity. Solid waste is really important because cellulose and derivatives of it are the most common form of biomass on Earth in terms of waste. And what we want to do is be able to process this. So if you imagine ethanol fuel, right? We have ethanol fuel that's derived from corn. People don't really like ethanol fuel that's derived from corn. It takes a lot of energy. It has a lot of waste involved. What if we could take all that leftover biomass, all of that leftover cellulosic stuff, and just convert that into hydrogen? And so what we want to find out is, can we use groups of bacteria, or what's called diverse communities of bacteria? Can they team up? Can they work together to couple the fermentation or the breakdown of solid wastes into current production and eventually hydrogen production in an MEC? And we know that there are a lot of bacteria that are thermophilic gram-positive firmicutes that can do this at heightened temperatures. And we know that they contain things called cellulosomes, which are actually organelles that bacteria have that are capable of breaking down cellulose. Some of them can also excrete enzymes that are cellulolytic that can break down cellulose. Now, if we have a microorganism that can break down cellulose into acids like acetate, and then a microorganism that can consume those acids to generate current or hydrogen, then we have the potential of converting solid waste into current or hydrogen in a microbial electrolysis cell. And so that's exactly what happened when we got a diverse community of bacteria and fed it only cellulose. We saw that cellulose was first fermented into lactate and acetate. And then after the lactate and acetate accumulated, it was consumed to generate current. And then I took the biomass from both the bulk, so what I would assume is where most of the fermentation is happening, and the biomass from the anode itself, which is where I would assume most of the respiration is happening. And I wanted to look at what bacteria are there and what could they possibly be doing. And so I'm just going to focus on the four big ones right now: the pink, the dark blue, the light blue, and the brown. And so in the bulk media, so this is where the biofilm isn't. So this is where the bacteria probably aren't going to need a metal. They probably aren't going to be performing respiration. They're probably going to be performing fermentation. And out here we see Brevibacillus and tepid microbium. And these two are essentially just bacteria that have been associated with degrading cellulosic materials and also proteinaceous materials. And I assume that these microorganisms are there because one of them is breaking down the cellulose that we're feeding it, and the other one is maybe breaking down some of the biomass that's decaying within this reactor. In the biofilm, I see Thermoanaerobacter and Peptococcaceae, which is actually most likely our Thermincola ferriacetica. And what we can see in this setting is that Thermoanaerobacter, Pseudoethanolicus, and Thermincola ferriacetica are working together in concert to generate current from the acids that these bacteria over here are producing. And the ultimate hope, the ultimate goal is that someday we'll be able to harness the energy that's in our waste in order to generate the power or the fuel that we need to happily live our lives. And with that, I'll ask if there's any questions. Okay. Okay, so those of us on the internet, the question is, and for all of you, if you didn't hear it, the question is why, if I found out the physiological stuff with Thermincola ferriacetica, why didn't I then do that same study with Thermincola pseudoethanolicus, which is a great point, and I would love to do that same research with Thermincola or Thermoanaerobacter pseudoethanolicus. It's kind of just a consequence of how the projects came to me, and what projects were being done at certain times and kind of the time overlap between the two. So it ended up that I was doing the kind of fundamental and physiological research with Thermincola ferriacetica, and then we had a visiting scholar, Kaser, come into the lab that was working with Thermoanaerobacter pseudoethanolicus, and I came over to kind of help that project out and ended up taking over, you know, a big portion of that project, and so it just kind of ended up that way. Mm-hmm. Yeah, so the question is, since Thermincola has multiple pathways, could that be an advantage? I mean, potentially it might make it more adaptive, but I don't know whether or not Thermoanaerobacter pseudoethanolicus has multiple pathways. It may very well. And from what it looks like now, a lot of microorganisms that we're studying, gram-negative and gram-positive, as I'm sure even Rachel can attest to, it's starting to look like they have multiple pathways. And I wouldn't be surprised if it ends up that most of the exoelectrogens or anode respiring bacteria that we're studying have multiple pathways, because a lot of them have multiple C-type cytochromes. Geobacter, for example, has over 70, 73. It depends on how you count them, but at least 70. And it seems like it would make sense to have if you have all those cytochromes around, that you would have multiple pathways that could adapt to changing conditions, different metal oxides that are around, things like that. Mm-hmm. Oh, we have a mic.
I wanted to ask you a question about that last slide where you showed the distribution of gram-positive and gram-negatives in your electrode.
Okay.
Where did those bacteria come from? Did you see them? Because I noticed there wasn't really much of a representation by the gram-negatives.
Yeah.
And was that just something that just happened, or did you select for gram-positives in that situation?
Yeah, so that reactor did have gram-negatives in it. I believe overall they made up less than 1% of the bacteria that were in there. And so what I mainly saw were gram-positives. You can go back to it.
So where did your seed come from for this?
Mostly from Firmicute. So I had serum bottles in the lab that were cellulolytic, so they were degrading cellulose at 60 degrees centigrade, or maybe it might have been at 55 degrees centigrade. And I looked at which serum bottles were performing the degradation the quickest, and I used those to inoculate it. And at the end, I just looked to see what was in there, and that was what was there. Is that 2 questions or a peace sign?
I think I have 3 questions, but
Oh,
depending on how long it takes to answer, maybe I'll stop at 2.
Okay.
The first is a question that I think I've asked you before, and I think we never got to a conclusion. So when you did your CVs at a low pH, you have a certain potential for acetate,
right,
but you're seeing current production begin even below the potential for acetate. So you're having current production at a potential where acetate could not act as an electron donor.
Right.
So do you have an idea of why?
Yeah. Yeah, I've thought about it a little bit too, because you've asked me this question before. And I don't know, it's kind of interesting. I mean, we've seen a similar phenomenon in other bacteria before. And, it might be, as we've discussed, the potential might be set in such a way that it's geared toward getting the electron outside as quickly as it can. So it might be a little more negative than the acetate itself. But it also might be, what's interesting about gram-positive bacteria is they have a high turnover rate of their peptidoglycan layer. And they can use this peptidoglycan, they can consume it, actually, some of them, and they can generate some energy for their cells. But I imagine also these biofilms are very, very, very thick. And so there might be decay in this biofilm. And so we might be actually looking at, you know, an electron donor that might be have a more negative potential than acetate itself, if that's what we're looking at.
Okay. And then my second and third, I think I'll join them together.
Okay.
So you showed one CV, a derivative, where you are modeling, I guess, both pathways as additive, right?
Yes.
You're adding the two. And this is exactly the opposite of how we've done it in Geobacter, where we thought that these were pathways that were shifting from one potential to other. As you shift the anode potential, you're actually shifting the use of the pathway.
Okay.
So my question is, have you tried fitting that CV in any ways to a model that does not have the additive pathways but separate pathways? And then related to this, the third question that I was going to ask was, from a lot of your CVs, it felt that you actually have 3 pathways, not 2.
Okay.
And so I just wanted your opinion on how you thought about these.
Yeah, definitely. I've thought about the idea of there potentially being 3 pathways, and that's very possible, especially given the size of that midpoint potential range. I mean, it's very, very large, but that depends too on which electrochemist you talk to. It's very possible that there could be more. I had that as a theory before at one of the, or I guess a hypothesis at this point, at one of the lectures that I gave prior to this. And for simplicity's sake, I have represented it here as 2. And I believe in the paper, it's written as 2 or more. But we know that there is more than just 1. And then what was the first half of that question? Oh, yeah. Yeah, whether additive is the best way to do it. And it's like, well, the thing is, if I did have 3 and it was somewhere right in the middle, would I even be able to tell using that model? And it's an interesting question to ask. It's something to think about too. And that is why originally I'd had, you know, when I presented this the first time, I had multiple hypotheses for how it's working, because that's very possible.
We have time for one more question.
Let me see.
Go in the back.
There are no questions online, so I guess this will be our last.
All right. So the end goal, making some sort of battery fuel cell to generate electricity. So, what are the risks of having parasitic bacteria getting in and consuming the nutrients without contributing to the process? So you have some that are breaking down the cellulose, but could you have non-electrophiles consuming the acetone and keeping your current generators from getting to the acetone?
Yeah, the acetate, yeah. Yeah. That's actually a major concern in a lot of our reactors, is we do have microorganisms, not necessarily parasitic bacteria, but we have microorganisms that get in called methanogens. They're archaea, and they can actually use some of those reduced organic compounds to produce methane. And methane, of course, is going to carry a lot of electrons, but that methane isn't going to be consumed to generate current or hydrogen. And you're essentially turning your anode into just a fermentation tank at that point. And essentially, yeah, your current, you're going to lose a lot of current doing that. And so that is an issue. That's one of the reasons for operating at higher temperatures. A lot of methanogens are more inhibited at those higher temperatures. I was trying to recently look at more cellulosic cultures, and what I saw as the cultures were degrading cellulose, in this case it was just compost, they're just degrading compost, producing a lot of hydrogen around 55 degrees. And when I bump that up, or sorry, a lot of hydrogen and some of them methane at 55 degrees. When I bump that up to 60 degrees, I didn't see any more cellulose degradation. And then again, I didn't see any more methane production either. So in that case, I think, yeah, what I had was a lot of methanogens, a lot of archaea that were in there. And so if we can get a system like this operating, as I've done, it might kind of help to weed out some of those methanogens anyway. And I guess that's the ultimate goal and the ultimate hope. We've also looked at, for example, Prathap in our lab has looked at specific ways that you can, and also Joe Myceli, who I don't see here today, they're also looking at specific ways where we can inhibit methanogens or prevent those electrons from being kind of, put into methane rather than current.
We're going to stop it here. So what's going to happen now is that the committee will stay with Brad asking questions right up until the time that Brad wants to go to 4 Peaks. So, so thank you for coming, and let's thank Brad again.
Thank you. Oh yeah.
Great. Thanks for having me.
Yeah. See you at 7, right?
I
You're going
already
to
have a prior commitment.
Oh, okay.
I'd love to be there.
Okay.
I'll take a rain check.
Okay.
All right?
Yeah.
Next time?
Yeah.
Beer's on you.
Okay. If you say so.
Good job.
Thank you.